Welcome to another session of the Science Talks broadcast program of the Turkish Scientific and Technological Research Council, Institute of Fundamental Sciences. Our guest today is a world-renowned researcher on proteins, in particular intrinsically disordered proteins. We welcome Dr. Vladimir Uversky. He is a professor at the Department of Molecular Medicine, Mursani College of Medicine, University of South Florida. He received his PhD degree in physics and mathematics. His particular field of study is biophysics at the Moscow Institute of Physics and Technology in 1991. And then he received a Doctor of Sciences degree in physics and mathematics again at the Institute of Experimental and Theoretical Biophysics of the Russian Academy of Sciences in 1998. Dr. Uversky has authored over 1,350 scientific publications, and he has been included in the prestigious Clarivate list of highly cited researchers every year since from 2014. In addition, in 2021, he was elected as a Fellow of the Royal Society of Biology and a Fellow of the Royal Society of Chemistry. And more recently, in 2024, he was elected as a Fellow of American Institute for Medical and Biological Engineering, as a Fellow of American Association for the Advancement of Science. And over the years, he has delivered hundreds of invited talks at various national and international seminars and conferences and workshops. And his speech today is going to be on intrinsically disordered proteins. And the title of the speech will be From Multifunctionality to Polypathogenicity with Intrinsic Disorder. We welcome Dr. Uversky, and we are eager to listen to your introduction to this exciting topic. Thank you.
So thank you very much for very kind introduction. So I cannot share my screen. I cannot see what should I do? I can see recording, I can see everything, but I cannot see how can I share my screen.
You should be able to share now. Yeah, you can, if you select the share button.
So there is no share on my screen.
There's an arrow in a yellow, in a green box.
Okay, now I can see it. Okay,
Yeah.
okay.
Yep.
Okay. Okay. So you can see it, yes?
Not yet.
How about now?
Okay, it's coming up
Yeah.
now. Yeah, it's we can see your presentation.
Okay. So before moving anywhere further, I will start once again with acknowledgements. So first of all, thank you for inviting me to give this talk here. And second of all, so you pointed out that I have quite a few papers, and quite a few of my papers will be coming from quite a few of my collaborators. And I'm starting with several slides, which I will not spend too much time on. So it's a list of my collaborators. It's not moving. Okay. It's my collaborators. For some time when I was making presentations, I was ending my presentation with acknowledgements, and then I decided I need not to end this stuff, but start with it, because I thought this is the most important part of my presentation. So definitely you can leave after this slide. So, and I was showing names, and then I decided I need to show faces. And I started to collect a set of people who was collaborating with me. And somebody told me that so it's a very nice collection. It looks at you, it's my kind of photo book of colleagues. And I was doing this stuff systematically for a very long time. I don't have any possibility to discuss who these people are, so why they are included. So, but all of them contributed somehow to my research, and all of them are crucial for my research one way or another. So so I was pretty successful till some particular moment in my life, which happened in 2016, So this my Facebook was actually hit very heavily so by this paper, which had almost 2,500 courses. So definitely now I cannot add pictures, but still had some hopes. And then I got another paper published. It's not moving. Published in where in 2019. So I had another 1,500 courses, and then I got another paper published in 21, right in the middle of COVID. So it's almost 3,000 courses. So altogether, I have a very large set of colleagues around the globe. So as you can see from the slide, I'm collaborating with very, very large number of different people in different countries and in different research institutions. So this is my interactome. So, and definitely, I wouldn't be able to represent all of the stuff which I will be showing today if it wouldn't be contribution of all of these people. So I'm definitely very thankful to all of them. And so three very important comments here. So if you do like what I'm talking about today, it's not me, it's all of these colleagues. If you don't like it, please don't blame them. It's not them, it's me. So and also keep in mind that I'm talking about intrinsically disordered proteins, which are known to be highly promiscuous binders. So they're very intensively involved in various types of collaborations. So so as you can see from my interactome, I'm highly disordered. So therefore, I'm not responsible for whatever I'm talking today. So jokes aside, so this is kind of outline of my talk. I will introduce intrinsic disorder very briefly. I will show you how intrinsic disorder can be found. We'll talk about very important concept, which is protein structure function continuum. And then we'll talk about involvement of disordered proteins in different diseases. And I will add to some conclusions. So introduction of intrinsic disorder is really very simple. So because, if you open any biochemistry textbook and if you start reading about proteins, what you will see this traditional classical structure function paradigm. So according to which amino acid sequence need to fold into 3D structure, so it should be responsible for unique protein function. And so this is a famous lock and key model, which was proposed by Emil Fischer very long time ago in 1893. And then it was amended to induced feed somewhere in the middle of last century. So, but overall idea here is actually very, very straightforward. So we have a very unique correlation in between amino acid sequence, protein structure, and protein function. So, so this is, looks like my computer, okay, it's moving some. Okay. So this is a very productive idea. So, personally, I'm considering it as a big bank of protein science. So because soon after it was proposed, people started to analyze this idea. And everything which we learned about proteins by the end of last century was heavily based on this intrinsic idea, on this protein structure function paradigm concept. So, but then I have next slide. This classical structure to function paradigm works generally, but somehow not for my proteins. And I have a set of crying boys and girls here, so which actually will be a reflection of my experience in conferences. When you start introducing intrinsic disorder to community, one of major features of intrinsic disorder, if you have highly disordered protein, it's not crystallizable. At least it's not easily crystallizable. So, as soon as I will be mentioning this point in my talk, after talks, I had a conversation with two girls and with one boy. Girls were crying, boy wasn't crying. So, but three of them were telling me in three different places. So they were telling me, so thank you for pointing out that proteins, not all proteins are crystallizable. So because I was working in structural biology lab, I was asked to crystallize protein, I failed. And I was told that I'm not a scientist because I cannot complete a very simple task. So what if it's not me? What if it's my protein? So and with very high probability, so they would be correct. So probably if you cannot crystallize protein using traditional set of conditions, so something is not wrong with you, something is not quite correct with protein sequence. Maybe it's not easily crystallizable. So, so these disordered proteins were recognized by example. And many people observe this stuff. And to me, a journey in intrinsic disorder started a long time ago. And actually, this is a very important slide, which is a reflection of my conserved stupidity. I would define this stuff in this way. In 1993, somebody came to my lab, colleague of mine, it still was Russia. And he brought a protein in a Eppendorf tube, and he said, I have a funny protein here. You can boil it a week, not only overnight, you can boil it a week, but as soon as it will be cooled down, it still will be active. And I cannot measure concentration of this protein. And I was looking at this guy and I was saying, so it's probably not very probable. Because is it protein from some thermophilic organism? And he told me no, so this is a human protein. So then I asked him, maybe it's not a protein, maybe it's a peptide, and he showed me this electrophoresis, gel electrophoresis, and as you can see, it's definitely not very large protein, but it's protein. If you're trying to measure concentration, so what you will do, you will have a solution of your protein, you will put the solution into test tube and put it in spectrophotometer, expecting to see absorbance around 270 to 80, so which will be corresponding to absorbance of aromatic residues. And as you can see, it's flat here, so you don't have any absorbance in this area. So definitely it's a protein which cannot absorb light, and it's not a peptide, and it's something very strange. And it's a protein not from thermophilic organism, but from human protein. And once again, it was 93, and it took me 6 years to recognize it. So it's not a unique, not very interesting exception, but it's a very interesting new set of proteins which need to be considered very seriously. So this protein is proteomycin alpha, and as you can see from amino acid sequence, it's extremely charged. So it has very strong charge repulsion. It doesn't have aromatic residues. It cannot fold because if it has strong charge repulsion, it doesn't have any hydrophobic attraction. Because if it doesn't have aromatic residues, it cannot absorb light. So from viewpoint of physics, it's a highly charged polypeptide, so which is not real protein. It's an exception, so it's not very interesting. It wasn't just me. Such kind of proteins were observed in literature for long time, in scientific community for a very long time, and so everybody was considering this stuff as an exception. So because people were extremely interested in structure-function correlation, and so definitely if you don't have structure, so something is not quite right. So long history of this field actually is reflected in different terms which were used in literature to describe these proteins. And we have kind of paleontology which is related to this phenomenon. And I'm showing this stuff in the next slide. So by the end of last century, so these proteins were known by all of these different names. So and definitely people who worked on vulnerable protein were not able to recognize it. So their proteins from this category of vulnerable proteins will be related to protein dancing proteins or to protein clouds or to 4D proteins. So what happened at the end of last century, a few groups simultaneously and independently came to ideas that so what if such kind of non-foldable proteins which are known as intrinsically disordered proteins, so what if they're not exceptions? What if they represent a new class of proteins which could be pretty abundant in nature? So and we start working on it, and it happens. So this is the case. And so I'm showing this very interesting and important slide, which represents the fate of polypeptide chain after biosynthesis in a cell. So for a very long time, belief was that so polypeptide chain must fold into the specific structure which will be responsible for specific function. Approximately 30 years ago, maybe a little bit more than 30 years ago, it started to be clear. And so you have also a very important pathway which is related to protein misfolding. So protein misfolding and aggregation, so were known for human community for very, very long time. So but this phenomenon was ignored. So because people were looking for structure, and protein misfolding and aggregation will not give you structure. And it's not interesting, it's an artifact. So 30 years ago, people recognized that misfolding is correlated to disease. So, and now you do have a possibility to get money for your research. So because you can claim that, so I'm analyzing molecular basis of disease because of this misfolding and aggregation is related to diseases. So now, in addition to this folding and misfolding pathways, we also have non-folding pathway. And so what you do know now that quite a few proteins, I'll show you in a few slides how many of those. So quite a few proteins, they're not folded, and they're not folded to different degree, and they're still biologically active. So what is also important, so none of these pathways will be a dead end. Folded protein can misfold, and actually it happens all the time in our body and start to be very visible with age. So because you have quite a few neurodegenerative diseases, so which are age-related, and so they will be originating from this misfolding, which is accumulating with age. So, but also misfolded proteins can be fold back into foldable proteins. So using chaperones and other metastasis machinery in a cell. So, intrinsically disordered proteins can misfold. Some of them can fold at least partially in order to become functional. So, but what is also important, structured proteins, so quite a few of them, so you'll have a very interesting feature. So they do have structure, but they're not active. So, and they need to be activated, and activation quite often will be associated with at least partial unfolding of these proteins. So it will be actually very important loop coming from order to disorder and coming back. But once again, all of this stuff running around, and that's actually very important feature, which actually makes analysis of proteins much more attractive and much more difficult, because we are not looking for structure, we are looking for everything now. So it will bring us now into abundance of intrinsic disorder. So when we just started working on these proteins, so we had this feeling that so it will be a few proteins which are intrinsically disordered, but not too many. So because if there will be too many, so why people didn't observe them before. So and we elaborated quite a few computational tools for predicting intrinsic disorder. So and what it was found that actually we analyzed archaea, bacteria, single cell eukaryotes, multicellular eukaryotes. So this is number of organisms which were analyzed 13 years ago. So this is a range of their proteomes. So this is an average proteome size. And so these three last columns will be three different measures of intrinsic disorder. First column is actually a very simple measure, how many residues in entire proteome are predicted to be disordered. And as you can see, so these values are ranging from about 10% to about 45, 50%. When we introduce this idea that we do have, say, 10% of proteins, 10% of residues are disordered, so we were told it's not very interesting. So because if you have 100 residues along protein and 10%, it means that so you have 10 residues if they are spread through entire sequence. So who cares? So what you need to look for, regions. So and as a result, we have the second column, which is shown here. That so we have so how many proteins in all of these proteomes. So we'll have long disordered regions. And long disordered region is defined as at least 30 residues in a row. And why we were using 30 here? So because in the universe of foldable proteins, so 30 residues is considered as a minimal foldable unit. So you cannot have natural protein below 30 residues which can fold. So typically it will not stay folded. So and as you can see, when we are adding these long disordered regions, so values are not getting lower, values are increasing. So once again, from about 10% to about 60, 70%. So definitely you cannot ignore them. But now we also have other computational tools. Which are not evaluating intrinsic disorder on per residue basis. So which are just telling you if your entire protein is expected to be structured or disordered. So it's kind of binary predictor, yes, no, structural disorder. And when we apply these tools, so to all of these proteomes, we have found that so from about 3% to about 40, 45% of proteins are expected to be completely disordered. So and so this last line here, so it will be characteristics of human proteome. And I claim that so I'm intrinsically disordered. And if you look at this picture, you can see that so not just me, all of us intrinsically disordered. So there is also an interesting observation that so amount of intrinsic disorder is increasing with complexity of organisms. And it's more reflected in this slide. So this will be viruses. and I don't have time to talk about this stuff. These are very interesting creatures, and they have lowest and highest amount of disorder in the entire protein universe. So what we're looking for this right part of slide. Green circles will be bacteria, blue circles will be archaea, brown square will be single cellular eukaryotes, and triangles will be pink triangles will be multicellular eukaryotes. And there's dependence of average fraction of disordered residues on proteome size. And as you can see, we do have actually one cloud which will represent simple organisms, and we have another cloud which will represent more complex organisms. So transition from prokaryotes to eukaryotes require not only increase in proteome size, not only increase in genome size, which is quite obvious. So because eukaryotic cells are much larger in comparison with bacterial cells, definitely you need to have much more coding material to fill these box with actual active and working features. So it's not only increase in proteome size, but it also increase in intrinsic disorder. So for us, more complex organisms to be more complex and be kind of more evolved, so we need to have much more intrinsic disorder. And from viewpoint of physics, it's an interesting observation because, you know, so complexity, which will be originating from lack of complexity, because the proteins are much less complex in comparison with structured proteins. They don't have structure, they have reduced sequences, reduced amino alphabet. So which means that so definitely it's something really very interesting is going on here. So and also because of it was pointed at the time physicist, I'm very pleased to see this picture. So from physical viewpoint, so we have a first order phase transition. So from simple organism to more complex organism. So we have one cloud which will be simple organism, and we have another cloud which will be more complex organism, and we almost don't have any points in between. So which means that so definitely in order to be more complex, so you need to have two features simultaneously, more disorder and larger sequences, larger genomes. So why we need intrinsic disorder? Why it's so abundant? So if it would be a few, so answer will be very simple. It's just fluke. It happened by chance, and it's junk. So because they don't have structure, so definitely we cannot take them seriously. And so people who were describing this disordered proteins before it started to be a very popular field, so quite a few of them were calling these proteins junk proteins. Actually, by analogy with non-coding pieces of DNA in human genome, so we have non-coding pieces of DNA which are junk DNA. So now we have also junk proteins. In reality, it's not the case because nature is a highly conservative lady, and if nature is generating something, it's for some purpose. If it's not useful, it will be very easily eliminated during evolution. So definitely intrinsic disorder is crucial, and it's crucial for protein functionality. So so this slide shows you how disorder is crucial for protein functionality. If disorder is not considered at all, we have classic structure function paradigm. So we have sequence which has a possibility to solve protein folding problem. We get specific structure, and it will be coming into specific function. Most often they will be associated with enzymatic activity. So if you allow protein to have intrinsic disorder at different stages of its life, so we can actually dramatically increase what proteins can do. So we do have conformational switches. So keep in mind that so this is actually an ancient slide which was designed in 2001. So at that time, so these conformational switches, so were actually very unique exceptions. There were only a few of them. So now people are talking about metamorphic proteins, and we have a kind of increasing population of these metamorphic proteins. And what does it mean? So we have one structure. It will be old order. And then we have a new structure, it will be new order. So, but in order to go from old order to new order, so you need to have a loop related to intrinsic to unfolding. So protein cannot kind of undergo transition from alpha structure to beta structure inside of folded chain. So it needs to be at least partially unfolded in order to do that. So another very important possibility is shown here. So we have quite a few proteins, as already pointed out, which are folded but not functional and need to be at least partially disordered in order to become functional. And this is crucial from my viewpoint, actually, even not from viewpoint of how protein acts, but also from philosophical viewpoint. So to make a very strong point that the disorder is crucial. So why it's so? Because so before the stuff started to be popular and it was introduced in a systematic way, people were considering so this bottom pathway, and as you can see, so some proteins cannot fold. Instead of staying in 3D structure, they will represent a flexible ensemble, so conformational ensemble, and so quite a few of these proteins will be able to undergo disorder to order transition. And they will get function because of this disorder to order transition, and this disorder to order transition will be induced by binding partners. So, and because of this pathway was here, people who were opposed an idea of intrinsic disorder, they were saying intrinsic disorder doesn't exist. So because, yeah, you have proteins which are waiting for partners, so, but they're not biologically active, and all biological activity still will be coming from structure because they will fall upon interaction, and so biological activity will be coming from this stuff, it will be coming from structure. So, and so by default, it's not a complete understanding and complete description of the system, because so disordered protein, so which is not folded under physiological conditions, at exactly same conditions at which folded protein is folded, so definitely doesn't have all information to fold. So it definitely missing something. So this missing information is added from binding partner. So and so disordered protein so is able to find so this binding partner is able to recognize it, is able to fold upon it, interact with it, and fold upon interaction. Which means that we have a few stages. So before giving the functional state, and this a few stages before still will be related to intrinsic disorder. But also so coming back to this upper pathway. So this disorder will require folding in order to be functional, but as I pointed out, we have structured proteins which need to be unfolded at least partially in order to become functional. Unfolded to become functional, which means that so definitely disorder is important. And in addition to all of this pathway, we also have another pathway, so which is shown that so we have quite a few protein functions which will be linked to lack of structure. So and once again, looking at this picture, if you will eliminate everything which is related to red, so protein functionality will be limited to upper pathway. If you add disorder, so which is shown here by red color, so it will tell you that so we have much larger possibility for protein to act. So intrinsic disorder is not comparable with catalysis. So this is bold statement, and it's really applicable to current world. So, but it's not very correct evolutionary wise. So because enzymes, which are catalysts, so they are a result of evolution. So, and definitely I don't believe that nature will generate a unique protein with highly sophisticated structure out of blue, as they will try to find function for this unique structure. So definitely first catalysts are were not very good catalysts. They were kind of very sloppy. And definitely they weren't very structured. So and but it was a good starting point. And coming back to a role of intrinsic disorder, disorder was crucial in evolution. It was starting point for everything. So but at this point in evolution, you don't have too many disorder within active sites of enzymes. Enzymes still will have disordered regions which will be used for different purposes, like regulation, recognition, and signaling. So, and why disorder is crucial for regulation, recognition, and signaling? So it's illustrated by a few slides. So first of all, so when you have protein which is unfolded, which can fold upon interaction with specific partner into specific structure, so you have so-called induced folding mechanism. So and it actually allows you to create complex which will be characterized by high specificity, so combined with low affinity. So which is really very crucial for signaling interactions because they are specific but not very tight. Signal interactions actually is on-off switch. It need to go up and down all the time. So these proteins can be involved in these polyelectrostatic interactions, or as I'm claiming, protein plane staccato. So doing such kind of interactions. What is going on here? So you actually, as illustrated by the slide, you can have a situation in which you have completely folded protein, which has an active site, so which is able to recognize phosphorylated serines and threonines in binding partners. Then you have binding partner, which is heavily phosphorylated and it's highly disordered. And each of 9 serines and threonines, which are present in this unfolded protein, so they have equal probability to interact with active site. And as a result, at any given moment, so we do have this touching approach, touching mechanism. So it's a real interaction. You can measure binding constant, and binding constant could be pretty high, so it could be pretty tight binding. So and also, but there is no fixing, so you don't have formation of stable complexes. Still will be doing such kind of interactions. But also, what is very important here, so strength of this interactions will be proportional to phosphorylation degree. So in this particular case, we have a protein which has 9 phosphorylated serines and threonines, and so it has strongest interaction. So when all 9 serines and threonines are phosphorylated, and if you'll decrease the stuff one by one, affinity will decrease. So before you will reach a kind of threshold of 5, below 5, mechanism, so complex will not be formed. So combining intrinsic disorder with phosphorylation will allow you to create a complex which will be pretty specific, and you can increase strength of this interaction. So in addition to plain staccato, some proteins can be involved in, so 2 disordered proteins can interact with each other, forming this 2 clouds touching each other mechanism. So we have induced folding, we have 1 disordered protein touching, palpating, so folded protein, we have 2 disordered proteins interacting with each other. So intrinsic disorder actually allows you to explain a lot of phenomena in biology which are not easily understandable from viewpoint of looking at the stuff for protein structure. So, and one of those phenomena will be alternative splicing. So, and alternative splicing is shown here. It's a complex mechanism which is present mostly in multicellular eukaryotes. It was invented by nature in order to increase proteome size, keeping genome size and what is going on. So, so in addition to coding regions, eukaryotic genes will have non-coding regions. And so from when DNA is converted to RNA, all non-coding regions will be removed. Protein will undergo, so mRNA will undergo normal splicing. So we have a canonical form of mRNA, so which will be translated into a canonical form of protein, which is supposed to fold into specific structure. So alternative splicing, it's a mechanism in which, in addition to all non-coding regions, some coding regions will be removed. Instead of full-length mRNA, we do have truncated mRNA, so which will be coding for truncated form of protein. So looking at this picture, so I have, instead of structure, I have this question mark here. Which actually a reflection of a very serious question. So what it will be consequences? So for protein, so for foldable protein, if part of sequence of foldable protein is taken off. Answer is actually not very simple. So you do have one possibility when such kind of protein will be able to survive if you have multi-domain protein. So you can remove one of domains, remaining domain will be able to fold. So you have a flexible loop on surface of protein. If you remove the star by alternative splicing, so remaining protein will survive. You can take a few residues from within the protein, so for protein global, but not too many. So typically, maybe no more than 5, maybe 6, protein will be able to survive. So in vast majority of other cases, if you'll take, as it's shown here, portion of foldable sequence, so protein will not fold. Foldable protein which cannot fold, so it will misfold, and protein misfolding is linked to disease. If it would be a unique event, which is not very common, probably it would not be a very large disaster. So in reality, alternative splicing is extremely common. So 75% of human proteins, human genes will undergo alternative splicing. So not all human genes will have intronic zone structure. So some of them will be just entire coding region. If you have this mosaic structure with zones and introns, 95% of such kind of genes will have alternative splicing. So now we have an issue. Most probable output for alternative splicing in the case of foldable protein will be protein with folding. And nature is using this stuff very, very intensively. So how it works and why it works. So why we don't have this conformational disaster, why we don't have this folding collapse, why we don't have immediate death. So because stuff which is alternatively spliced in mRNA is mostly coding for intrinsic disorder. So you can take any piece of null structure, remaining null structure still will be null structure. So it won't be affected much. So, but it will change dramatically how protein acts. So because as it's shown here using breast cancer protein 1 as an example, so this protein has a very long disordered region in the middle, so which is involved in multiple interactions, so which has a lot of post-threshold modifications. So so this light blue bars show different isoforms of this protein. Canonical is a full length, and dashed lines will show regions which are taken out by alternative splicing. So, and now shaded areas, I will show you which functions will be removed. So by alternative splicing. So looking at this picture, it's quite clear that alternative splicing is really very crucial. So because what it does, it helps protein to avoid collapse, and it helps proteins to increase functional repertoire, so and still survive and do a lot of very useful stuff. Another question will be correlation between intrinsic disorder and post-threshold modifications. And a very nice example will be given by phosphorylation. So everybody knows about phosphorylation. So there are 520, maybe 550 kinases. And when I was talking to people who are systematically working on phosphorylation, when I asked them how common phosphorylation is and how many proteins, for example, in human will be phosphorylated, I got actually a very, very bold answer. Each protein will be phosphorylated at least once during its life. So many proteins can be phosphorylated at multiple sites. So, and now we have another issue. So we have 520 protein kinases, and we have at least 20,000, 25,000 canonical forms of proteins, so which can be phosphorylated, and some of them can be phosphorylated at multiple sites. So we definitely don't have one-to-one ratio which you would expect for lock and key model. So you have one lock with multiple keys, and with very high probability, it will not be rigid key. It will be flexible lockpick. So the intrinsically disordered regions will be used in order to hold phosphorylatable sites. So it is crucial from this point. So if you have an issue here with kinases, so you have larger issue with phosphatases because so you have just 250 phosphatases. So so phosphorylation is a reversible process. Everything which is phosphorylated will be dephosphorylated. And so once again, for each phosphatase, we do have quite a few potential targets. So, and probably I'll skip this stuff because I just mentioned it. And it's not only phosphorylation, it's looks at it's any type of post-threshold modifications which are catalyzed by specific enzymes. So if you have something which needs to be modified, so modifier with very high probability will be structured. So, but something which is modified, so will be intrinsically disordered or will be included in intrinsically disordered regions. So, because disorder is crucial, so for binding and recognition and being able to adjust to active site of enzymes. Last example here will be protein-protein interaction networks, so which are very complex. So definitely, if you look at this picture. So this will be protein-protein interaction network. Each point here will be protein, and each line will be protein-protein interaction. And as you can see, we have quite a few sunflowers. So meaning, so we have quite a few proteins which have very large number of potential binding partners. So this will be hub proteins. So and the presence of hubness in protein-protein interaction networks, once again, is not compatible with lock and key. So because in case of half, so one protein will be able to interact with not a few, not quite a few, but quite often with a very large number of different binding partners. So it could be a few thousand. So which means that so it's not definitely a rigid body interaction. Something is quite different. And so when we analyze the stuff, we have found that so half proteins, quite a few of them will be highly disordered. Some half proteins could be structured, so but their partners will be intrinsically disordered. And then you have a combination of these two possibilities: half protein and half protein partners also both will be disordered. So and binding promiscuity so will be so kind of driven by intrinsic disorder. So and it's further illustrated by P53. So everybody knows about P53. So it's the most studied protein. So if you go to PDB and if you look for structures of P53, you will have very large set of structures. And the vast majority of them will be looking like what I'm showing here. We have DNA binding domains, which is predicted to be structured, which is structured, which can interact with binding partners. So, but also you have about 60% of protein which are expected to be disordered and been shown by experiment to be disordered. So, and different pieces of these disordered regions, so you'll be able to interact with binding partners. So, vast majority of interactions will be originated from intrinsic disorder, and different pieces of protein can be used for these interactions. It's actually one-to-many binding scenario. And I do like this C-terminal part of P53 because actually it shows a very, very cool mechanism here. So exactly same region is able to interact with at least 4 different binding partners, and it falls in 4 different structures upon interaction with binding partners. And it's actually, from my viewpoint, a final counter-argument. So for validity of statement, one sequence, one structure, one function, because we have one sequence, multiple structures, and multiple functions. And so this is really very important because this morphine structures in C-terminal part of P53, so will allow this protein to regulate at least 4 different pathways. So P53 is a crucial player. So so it's actually does this stuff because it has a lot of disorder, so which is pliable and multifunctional. And it brings us into protein structure function continuum, so which is really very crucial. So we are first looking at protein structural heterogeneity of IDPs, so which can be described as a spectroscopy of intrinsic disorder. Why I'm talking about spectroscopy because of this slide. Before intrinsic disorder was introduced at the end of last century, protein universe was blue. All proteins were structured, all proteins were rigid, all proteins were cold, not moving. When we start introducing intrinsic disorder, so we were intentionally looking for proteins without any structure, but functional. Why it's important? Because if you are fighting with 100 years old paradigm about structure and function, so you need to have complete lack of structure and function. So in this case, it will work. If you have a protein which is a mixture of structure and disorder and is still functional, so people who doesn't believe in disorder would say disorder is garbage. So you have still structured pieces which are responsible for functionality. Therefore, originally we were looking for these noodles. So protein universe was split on blue and red. Almost immediately when we start kind of looking at the stuff more intensively, we recognize that this blue and red or black and white is not correct. So because what we have instead, so we have this continuous spectrum in which entire protein can be disordered to different degree, different parts of protein can be disordered to different degree. We have a very, very wide spectrum of structural features for proteins. It's not just structured and completely disordered, it's kind of structured and disordered and everything in between. So as a result, so we have this idea that proteins will have kind of differently structured pieces. So proteins will have fold-ons, and fold-on it will be independent foldable unit, so which is not a domain. So single domain protein might have a few fold-ons, it's smaller than domain. We can have inducible fold-ons. It will be regions which will be able to, which are disordered, but will be able to hold upon interaction with binding partners. It's like an N-terminal part of P53. We have inducible morphine foldons, so illustrated by C-terminal part of P53, in which region can fold differently upon interaction with different partners. So we have non-foldons. So this is non-foldable regions or non-foldable proteins, so but still biologically active. So semi-foldons, so which are kind of once again semi-folded all the time. And then we have also very important feature, which is unfoldons, regions which are structured but not active and then need to be unfolded in order to become functional. So if you put all of this stuff together, so you can recognize that so all of these foldons, unfoldons, and different type of foldons and semi-foldons and non-foldons, so it can be present within one protein, and all of these pieces could be responsible for different functionality, which immediately tells you that so in one protein it's not one, it's definitely one sequence, which is not also quite correct in a second. So, but it's also quite a few different functions. You don't have kind of 1-to-1-to-1 ratio which you need to expect. So people did recognize also that so majority of complexity of biological systems will be coming from proteins, but not from genomes. So, for example, in human genome, so we have 21 protein encoding genes, 21,000 protein encoding genes, or maybe 25,000 protein encoding genes. So, but we have a very large number of different proteins. So and as it shown here, it could be more than 1 million, so which is not quite correct. I think it's a kind of very, very low ball. So because so even for single protein, which is tyrosine, which is the longest protein in our body, for tyrosine, so it has about 50,000 residues. It has a very sophisticated internal exon structure, and it has about 1 million alternative spliced isoforms. So, which means that so from one gene, so you can generate a very large number of different forms, which are called, which are known as proteoforms, and so it will be originating from alternative splicing, from phosphorylation modification, from intrinsic disorder. So, and as a result, instead of classical understanding one gene, one polypeptide chain, unique structure, one function, so we have this red picture here. Well, from one gene, we can have multiple polypeptide chains, so which will be folded or not folded to a different degree, or folded to a different degree. And we have very large set of different functions which will be coming from such kind of stuff. So, and once again, coming back to P53, I'll skip this stuff. So coming back to P53, so we do have so this firework representation of protein structure function continuum. So we have one P53 gene. So because of phosphorylation modification, mutations, alternative splicing, so we have multiple proteoforms, and we have multiple combinations of all of these features. And as a result, we have this full whole sky. So with this firework, so it will be reflecting what this protein can do. So, intrinsic disorder is also very crucial. So from the point of liquid-liquid phase separation and liquid-liquid phase transition and formation of membranous organelles. So this is a very cool, very hot subject which started to be extremely attractive to researchers within last maybe 7, maybe about 10 years. So cells now could be considered as aqueous multi-phase systems. So what does it mean, liquid-liquid phase transition? Liquid phase separation. So under some conditions, you have a homogeneous mixture, so which will contain all of components which are evenly spread within volume. Something could happen, and now you do have this liquid phase transition, as a result of which you have formation of droplets. So what is crucial here? So this will be liquid droplet located in liquid. So it's water in water, and it doesn't have any embedded membrane. So all of the stuff is purely based on polymer physics, purely based on interaction of polymer with itself and with environment. And as it's shown here, so this transition can be triggered by temperature, concentration, pH, salinity, and whatever you can imagine. And so it also will be regulated, it can be regulated by conformational flexibility, phosphorylation modification, sequence simplicity, multivalency, and modularity. So what is also important, so this is a response of a system to change in environment, and it's a phase transition. So what does it mean, phase transition? So it's like in this way. So something changed and you have phase separation. Something changed back and phase separation is gone. And this is actually, so this trick is an explanation why people do not pay too much attention. So because we are using in science a reductionist approach. So we have a complex system, so we are trying to pull this stuff apart. We can take these parts and we try to understand how these parts work. If you have organelles, traditional organelles, so which are embedded in membrane, so you can isolate them, you can purify them, you can work with them. If you have this liquid droplet which is appearing and disappearing, so you cannot purify it, you cannot isolate it. So which means, so people didn't pay too much attention to it, assuming it's not very natural. Which is actually a very interesting point here. So because one of the most famous, so this membranous organelles, so will be nucleolus. Nucleolus was discovered well before discovery of all cellular organelles. So it was detected early 17th century, but people do not pay too much attention to all of this stuff because they couldn't purify it. Now it's recognized. So we do have not one, not two. So we have very large number of these membranous organelles. So which will be a result of the lipid phase separation. And formation of these membranous organelles is really very crucial. And people are starting to talk about this stuff as a probably major principle of how cell works. So you do have formation of all of these membranous organelles, specific compartments, so specifically populated with specific proteins, so which could be related to how everything is controlled and regulated in a cell. So this slide actually shows how common these membranous organelles are. So when they were kind of brought back to attention of scientific community somewhere around 2009, 2010, so there were not too many. In the first paper which we published somewhere around 2015, so we found about 20 described in literature. So this paper, which was published in 2018, so it has about 60. And a couple of years back, we published a chapter which described 110. Which means so we do have very large number of these membranous organelles, and people are discovering this stuff in a very regular basis. And I think it's not 110, it's in reality, it's much more. What is crucial about this stuff? So it's a liquid inside of liquid. It has all features of liquids. It's not embedded into membrane. It's easily accessible to outside. Anything can go in, anything can go out. So intrinsic disorder is crucial for their formation. So because it's not formation of complex in this way, it's formation of complex in this way. And when we're talking about such kind of interactions, it will be related to intrinsic disorder. So we analyzed abundance of intrinsic disorder in different membranous organelles. So in 2018, and we have found that all of them, so we have a very high proportion of intrinsic disorder. So probably I can skip this stuff also. And let's skip this stuff, and we'll be coming back to this. So I'm in Florida. So something in the water. How these membranous organelles will be originating, and so not because of alligators. So they are in water. So but also I have next slide. So these are also alligators at night, and as you can see, they have this ice reflecting light in different color. And why it's cool here, because so this is how membranous organelles will look like. So definitely, so there is some resemblance with alligators here. So, but what is important? So membranous organelle, formation will be related to how polymer will react with water, and how water will react with polymer. And it's not a real polymer or protein or protein RNA interaction. It will be interaction of protein RNA through water with each other. So we have some cool analysis done, and we do know for sure that water structure is changing. So when you're adding polymers, which is obvious, yes. So definitely this stuff needs to be taken into account very, very seriously, because this is how it works. All of other features which people are describing in literature, so that we have stickers and we have linkers, all of the stickers and linkers which are involved in the formation of transient multivalent interactions. so there will be secondary. So primary stuff will be water. So we need to look at water very seriously. So, and moving into, so how much time do I have?
Yeah, you can continue, until the end.
Okay, so
That's fine, no problem.
Okay. So, now, we do have intrinsic disorder, which is extremely common in cell, so which has a lot of functions, which means that so potentially intrinsically disordered proteins need to be tightly controlled, so which is quite obvious. And very cool analysis was done. So once again, so pretty long time ago, and what it was done here, so computationally. People looked at what it will be a difference in between structured proteins and disordered proteins in a cell at different stages of protein life. And what was found, so first of all, structured proteins and disordered proteins are encoded differently in DNA. So which is quite clear. Yes, so if you have highly abundant presence of some residues and not so abundant presence of other residues, so the stuff should be reflected somehow at DNA level. So what happened? So intrinsic disorder is more prevalently encoded by CG enriched DNA and RNA. So we have different encoding mechanisms. We have different translation rates. We have different transcription rates. So we have different stability of mRNA. So we have different stability of proteins. So typically, when we have disordered protein, so it's considered to be not stable biologically because it's highly flexible. It's easily accessible to proteases. It could be digested and modified in a very simple way. So those will be kind of proteins with shortest lifetime, so will be intrinsically disordered. So, but proteins with longest lifetime also intrinsically disordered. So this longevity is coming from ability of these proteins to be involved in specific interactions and information of very specific complexes. So for example, so when two ordered proteins are interacting, so it's interaction in between two of my fists. When two disordered proteins are interacting, it will be interacting in forming such kind of complex. So and definitely such kind of complexes will be much more stable. And a very good example, one of examples will be given by elastin. It's highly disordered protein, so which doesn't exist as a monomer, which always form bundles, and bundles of elastin have lifetime of 70 years. Example could be even further enhanced by an observation which was made a few years back. Scientists excavated a skull from a bog in UK, and this skull was about 4,000 years old, and skull contained brain. So they have a piece of brain which was 4,000 years old, and they did proteomic analysis of this brain, and they have found that there's a high population of disordered proteins which sustained this 4,000 years of sitting in a bog. Which means that definitely disorder is highly chewable if it's by itself, but also will create a highly sophisticated and very stable structures. So everything is under control. Structured proteins and disordered proteins are controlled in different way. So which is a reflection of importance of disorder. Yes, if they're controlled in different ways, so definitely if disordered proteins are controlled in different ways, this will be crucial for what is going on over there. So if proteins are not controlled, now you do have a disaster. So you have this D-square concept, disorder in disorder or intrinsic disorder in human diseases. Disorder proteins are highly regulated. So if they're not regulated, so then you have all of these issues. And you have several genetic factors, and you have several non-genetic factors which will be related to this distorted regulation. So we have mutations, we have chromosomal translocations, we have alternative splicing. So we have difference in expression. That will be genetic factors, and non-genetic factors will be catalytic degradation, phosphorylated modification, and trafficking. These proteins need to be in right place at right time. So if they're brought in wrong place at wrong time, so you can have a disaster. All of the stuff will be played a role. So chromosomal translocation is when you have this stuff, you have two original genes, and because of some genetic aberration, now you do have this hybrids. So you have this hybrid protein in which one half of one protein will be combined with half of another protein, and vice versa. So this chromosomal translocation is a very common mechanism in quite a few of cancers. So there is a very large set of very, very bad early onset childhood cancers. All of them are kind of extremely bad, so the kids don't have a chance to survive. And all of them, or many of them, will be related to this chromosomal translocation mechanism. So alternative splicing. So if everything is nice, you have active and happy protein. If alternative splicing is screwed, so then you have either unhappy protein or protein which is kind of misfolding and it's not active. It's not good. So post-translational modifications. So it will be correlation between tau protein and Alzheimer's disease will be a very good example. So highly disordered protein tau. And I'll talk about this stuff once again in a second. So it has a few phosphorylation sites, so which are crucial for its interaction with microtubule. So if it's over-phosphorylated, because it has very large number of potential phosphorylation sites, so instead of being involved in regulation of microtubule assembly and disassembly, it fails off microtubule start to aggregate by itself and start forming these aggregates, so which are related to Alzheimer's disease. So stuff related to aberrant proteolytic degradation will be amyloid beta. So everybody knows that amyloid beta is related to Alzheimer's disease. Amyloid beta is a peptide which is derived from amyloid precursor protein. And if something is not regulated in the right way, so you have production of too much of this short peptides, they start to form aggregates. So and then we have development of Alzheimer's disease. So a few more words about a very cool example related to Parkinson's disease and related to alpha-synuclein. Parkinson's disease is second most common neurodegenerative disease. It's an environmental disease, which means that exposure to some particular components in the environment will make people more predisposed to have Parkinson's disease. And there is a very large set of very interesting data showing a correlation between exposure to pesticides, metals, or some components of drugs, of heroin, which will be causing development of Parkinson's disease. It's associated with death of the dopaminergic neurons in substantia nigra. Substantia nigra is a small part of middle brain. I'll show a picture in a second. And surviving neurons will have specific deposits as the Lewy bodies and Lewy neurites. So this is a PET scan of normal brain, and this is activity of substantia nigra. So you can see these yellow points, so which are reflecting full activity of substantia nigra. Substantia nigra is a place which is producing dopamine. So it contains dopaminergic neurons. In Parkinson's disease, as you can see, so definitely is not so much activity anymore. How the stuff is reflected if you're looking on slices of brain. Normal brain, so you have this substantia nigra, so the black matter located in the middle of brain. And so this portion is almost completely empty in Parkinson's disease. You don't have this black matter. And blackness of this matter is defined by these dopaminergic neurons. So they're producing dopamine. Dopamine is easily oxidizable by air, and as a result, it produces brownish color. So you don't have too many such kind of neurons in Parkinson's disease. The neurons which survived, so they will have these inclusions which are known as Lewy bodies. This will be aggregates. And these aggregates will be originating from alpha-synuclein. In addition to Lewy bodies, which are located inside of neurons, we also have aggregates of alpha-synuclein which are located in axons, and they are known as Lewy neurites. What is actually very interesting is not only Parkinson's disease. I'll show you next slide to confirm this stuff. It's quite a few other neurodegenerative diseases which will be related to misbehavior of alpha-synuclein and accumulation of alpha-synuclein. So in case of Parkinson's disease, so alpha-synuclein is accumulating in substantia nigra and misbehaving in substantia nigra. If it will start to misbehave in other different regions of brain, so it will develop all of the different diseases. And so in 1997, a correlation between synuclein and Parkinson's disease was found. And soon after, people recognized it's not only Parkinson's disease, we have a very large set of synucleinopathies, so which will be originating from misbehavior of alpha-synuclein. And so this is actually a really very nice example of polypathogenicity. So originating from multifunctional protein. So and a few more words about alpha-synuclein. It has alternatively spliced isoforms, as it's shown here. So it has possibility to interact with very large number of partners. I will show this stuff in a second more. It has multiple post-threshold modifications. So it definitely has all of the features which you can expect for disordered protein. It's predicted to be disordered by all computational tools which you can use. It was shown by experiment to be disordered, so highly disordered protein. It's another way to look at the stuff. Using this D2P2 platform, which is kind of a very cool representation of multiple disorder predictors combined with functional features of protein, presence of disorder-based binding sites, molecular recognition features, presence of different post-threshold modifications. This protein is extremely prone to phase separation, as it's shown here by analysis by push drop. So it has a high potential for phase separation and can phase separate. It was shown to phase separate. And it definitely acts as a protein hub. It has not a few, not quite a few, but a lot of binding partners. So people established that we have more than 500 physical interactors with alpha-synuclein. So definitely it tells you if you have something which can affect 500 partners, so you can expect that it can have a very bad consequences if it's not behaving in the right way. And it's a protein chameleon. This idea of protein chameleon was actually developed based on behavior of alpha-synuclein. So this protein is unfolded, but it can fold very differently in very different environments. And can fold very differently upon interaction with different partners. Which means that you definitely do have a high pliability and adjustability of this protein, and also will be related to why it's so bad from the point of being engaged in multiple diseases. What is cool? Up to now, as you can see, we have quite a few papers. Talking about alpha-synuclein, it was actually published in 2017. It continued to have exponential growth. We have more than 16,000 papers talking about alpha-synuclein now. But all of these papers, will be starting with a very interesting claim. Alpha-synuclein is a small presynaptic, intrinsically disordered protein with unknown function. And more than half of these papers, we'll be talking about one function or another function of this protein. How come protein which has so many functions is still assigned as protein with unknown function? And answer will be very simple. All of us were trained in structure function paradigm. All of us were trained in requirement for a unique protein to have unique function. And if protein doesn't have unique function, function is unknown. If it doesn't have prevalent function. Next slide actually shows you what is going on. This is actual NMR structure of alpha-synuclein bound to a vesicle, and it looks like this octopus. It has multiple arms because it has a lot of disorder, and because of this flexibility of its arms, it can do a lot, and it can oligomerize, and then you have possibility to do a little bit more and more and more depending on how the stuff is assembled. Intrinsically disordered protein, definitely has a lot of proteoforms originating from alternative splicing, post-threshold modifications, interactions from intrinsic disorder, And if you take this stuff into account, this multifunctionality of protein, so we'll be propagating into polypathogenicity of this protein. So definitely it will be coming back to. So when you have a protein which can do a lot, and it's not only can do a lot, it's also important point. So it can be present. So actually, alpha-synuclein is it's one of the most highly abundant soluble protein in the brain. It is account for about 1% of soluble protein. So it's very common. It has a lot of different functions, and definitely if it's not behaving in the right way, so we have a lot of issues. A few kind of closing slides we'll be talking about that it's not only alpha-synuclein. So just look at these three major players related to neurodegeneration: amyloid beta and tau protein. So I just discussed alpha-synuclein. Same stuff will be applicable to tau. It's not only Alzheimer's disease. We have tauopathies, which will be different neurodegenerative disease related to the behavior of tau protein. It's predicted to be disordered. It has a lot of disorder-based body insights, has a lot of post-translational modifications. So it has a lot of alternatively spliced isoforms. It has a very high tendency for phase separation. It's shown to be disordered by experiment. It's shown to be disordered by AlphaFold. It's a protein hub. So it's definitely involved in multiple interactions. Same is applicable to amyloid beta. It's not only Alzheimer's disease. There are quite a few other diseases which are known as amyloidopathies. It's difficult to work with this peptide. It's extremely hydrophobic. So computationally, when this analysis was done, it was shown that it does exist as a very kind of dynamic informational ensemble. And all of the stuff will be coming back on how this multifunctionality and ability to form different proteoforms and different oligomeric forms, how the stuff can be propagated into different types of aggregation, as it's shown here. So you can have association from monomers, which will have different shapes, and they can assemble in different way, and they propagate into specific oligomers, and the stuff can be propagated into different types of aggregates. In addition to ability of protein to be engaged in multiple useful interactions and multiple useful functions, so because of intrinsic disorder and because of interactability, you can also generate a lot of different aggregates, and all of these aggregates will be playing very different roles. And all of this is really very crucial because people do recognize that so when you have aggregates in proteins, so you have multiple ways of how cell can die because of presence of all of these different aggregates. Probably a couple of last slides will be related to how this proteinopathies will be related to membrane-less organelles. People did recognize now, as I pointed out, alpha-synuclein, tau, amyloid beta, all of them will show high propensity for liquid-liquid phase separation. So what does it mean? So these proteins will be easily able to form these droplets. And formation of droplet is a protective mechanism because many membraneless organelles will be originating in response to change in environment. So one example will be stress granules, but it's not only stress granules, it's a lot of other different examples. Typically membraneless organelle is a response to change in environment. It's supposed to stay in specific state for specific time, and then when environments change back, it needs to be disassembled. If it will stay for longer time than expected, what it will be, so you have a droplet which has a high concentration of aggregation from protein. So definitely now protein start to aggregate, and you have a very important initiating point for protein aggregation and amyloid fiber formation. So this is a new development in neurodegeneration. People are saying that membraneless organelles, droplets or biomolecular condensates, which are formed by these neurodegeneration-related proteins. So it will be probably starting point for neurodegeneration. Intrinsic disorder is definitely a very useful concept in protein universe, in protein field, protein science, because it allows us to answer many questions which cannot be answered using structure. And it's not in the way that intrinsic disorder is better. than structure. So in reality, they're complementary. So stuff which is accessible to structured proteins is not accessible to disordered proteins, and vice versa. We do have a very important combination between structure and disorder. So, and I think I'm done now. So we have highly abundant disordered proteins. So disorder is crucial for protein functionality and related to structure, function, and continuum. So disordered proteins are highly controlled. So if they're not controlled, so we have multiple issues. So we have very important correlation between disorder and function, disorder and membranous organelles, disorder and pathology. And definitely this stuff need to be taken into account very seriously. So, and thank you. So,
Thank you, Vladimir, for this exciting presentation. I have a few questions. I'm sure Orkide, also will have some questions.
Yeah.
In the classical, static understanding of protein structure, we have the sequence and then the shape, and then that translates to a certain function. So in the case of IDPs, we have in between an ensemble of conformations, right? Or is it more like a continuum? Or are there discrete set of conformations that are functional?
It's an outstanding question, and so there is no straight answer to it. So because in order to provide straight answer, so you need to have good examples showing in one way or another way. So it's a lot of examples which are showing also disordered proteins are conformational ensembles. So now question which will be coming back to your question. How many members in this ensemble are? So unfortunately, there is no experimental answer to this question. What we do have something like that. So it's moving all the time, and we have a possibility to have snapshots. And so people who are doing trying to find a correlation between experimental constraints which are described as this ensemble. With some models which can be used in order to generate ensemble. So the question was how many different members need to be taken of ensemble need to be taken into account in order to honestly represent ensemble. And not too many proteins were analyzed from this viewpoint. So, but the proteins which were analyzed, for them it was shown you need to have a few thousand. So, a few thousand, it sounds a lot. So from your point of one structure, one function, yes, on one side. On another side, you can imagine, so you have not a few thousand, you have not even a million, you have unlimited number of possibilities. So which means that potentially you have some limitation here. Also keep in mind, so disorder is highly heterogeneous. So which means that so you can have some proteins which will be more disordered, some which will be less disordered, and all of the stuff will be adding some additional constraints. And then another interesting question. If you are talking to a structural biologist and you're describing a protein, I have a protein which has 10% of which has alpha helical structure, so which is present 10% of time. So how you will take this protein? Is it structured or disordered? And structural biologist will tell you. So, sure, it's disordered. So it has, so 10% of sequence, 10% of time, it's nothing. So because it's not real. If you talk to somebody who is working with intrinsic disorders, they will say, oh, it's pretty structured because you have 10% of sequence, which is spending 10% of time on alpha helix. It's definitely something. It's not completely random. So also because of its polypeptide. And because of polypeptide has specific restrictions of what type of interactions can be present inside of polypeptide. You don't have, I think, infinite number of potential secondary structure elements. So because of geometry of polypeptide bond, so you have a polypeptide chain, you have actually very limited possibilities of how the stuff can be ordered part-time or full-time. So which means that so it's definitely not infinite number, it's limited number. So, and also, as I pointed out, people start to pay attention to this metamorphic proteins. And metamorphic protein is not a disordered protein, it's a protein which has different stable structures at slightly different conditions. It's not chameleon as alpha-synuclein because it starts from completely disordered state. So metamorphic proteins will have this capability of being folded very differently. So it's actually a lot of possibilities.
Well, the reason I asked this question is there are, as you know, AI-related approaches like AlphaFold, to predict the structure of a protein. It seems they are very successful in the case where the conformation is static. But if it is like an intrinsically disordered protein, should one seek a time-dependent continuum of conformations or discrete set of conformations?
Outstanding question. Very, very good question. And AlphaFold is hallucinating. AlphaFold. So I read this very interesting description. What does it mean? It's machine learning based technique. Yes.
Yes.
What does it mean? It can find something which was observed. It can find something which has some roots in what was observed. But it will not be able to find something which wasn't observed. And instead of telling, I don't know, it will show you something. How the stuff will be reflected, I'm using AlphaFold not very often, not in very systematic way, but actually pretty regularly, especially for disordered proteins. And what you can see for disordered proteins, you can see absolutely physically unrealistic structures. So for example, quite a few disordered proteins will show lone alpha helix, not supported by anything else. And lone alpha helix, it could be probably 300 residues long. So do you believe that you can have blue alpha helix staying alone in water? So there is no any physical reason for the stuff to be in this way. But AlphaFold is telling you that this is the way. So other issue with AlphaFold, it's misleading by giving you a snapshot. It gives you just one structure out of many different possibilities. Very logical and very nice development will be to generate conformational ensembles, and that will be actually equivalent to what is going on between X-ray crystallography and what is going on with NMR. So definitely, I do believe NMR more than X-ray crystallography because it shows that it's moving. I'm a physicist who is working in biology. Okay, and so in biology, it's very, very simple definition. So what is definition of life? So alive is moving. So if it's not moving, it's not alive. And if you convert the stuff back to proteins, so if you have your crystal structure, if you have your rigid rock, potentially it can do something. But not much. You definitely need to add flexibility over there. But I'm not sure that too many people are talking about this very large issue of AlphaFold, giving you just one member of ensemble. So because for a person who is not familiar with technique and who is just looking for structure, I have my structure which I can use for all of this magic related to structure-based whatever is there. But it's one of many, and especially if it has intrinsic disorder, which is shown as large blue alpha helix, I'm sorry, you are doomed. So you will not be able to get anything out of it.
So I have a question. Can I ask the question too?
Sure, of course, please.
So, as you know, Volodya, we are getting ready for the CASP competition next year. And there's a special interest group in CASP that meets every month.
Mm-hmm.
It's about conformational assembles and IDPs.
Mm-hmm.
And so we developed a new MSA, a new AI algorithm that considers IDP specifically and differently.
Mm-hmm.
But how shall we validate our results, and how shall CASP competition validate our results? PPE data bank, is that enough? What do you think?
So this plot will be one of possibilities. So, but this plot is outdated. It's not kind of not keeping on time. So PDB can tell you the structure is absent. So, but it will not tell you much more than that. So, and also coming back to validity of AI stuff, you know, so when people start to work on structural genomic initiative, so do you know what I'm talking about, structural genomic initiative? So, we will crystallize everything and we'll get all of the structures. So, and they immediately recognize that, so you don't want to do it because there is no reason to crystallize a sequence which has similar sequence to something which was already crystallized, because with very high probability you will have similar structure. What you need to do, you need to find sequences so which don't have structures.
Yes.
So, and they failed at the beginning. So because vast majority of sequences which don't have known structures will be intrinsically disordered. And intrinsic disorder is not heavily evolutionary conserved. So you have only a few small pieces of disorder proteins which could need to be conserved, and you have a lot of possibilities for variability. So, so which means that how to take this stuff into account? It's definitely need to be taken into account. So, but you don't have a solid platform for claiming that my disorder predictor is much better than your disorder predictor because my conformational ensemble is much closer to reality. So because we don't have actual description of reality as of yet.
Yes.
So
exactly.
So, but definitely, so you can discriminate between structure and disorder, better or worse. So, but also keep in mind that disorder is condition dependent.
Yes,
much more condition dependent on structure. So
yes.
what I'm telling to my students, so when you have structured protein, so you have this funnel, you have structured protein, it's a ball in the bottom of this funnel, and you're pushing this ball, so your environmental changes, and it will be shaking back and forth, and will be ending up in exactly the same position on a closed position in the bottom of this funnel. The protein is something just located on the top of the hill. You're pushing it from one direction with one strength, it will end up in one valley. It will be ending in another valley. So you do have extreme sensitivity to the environment. So which is actually not taken seriously into account by any predictor. So
I agree.
we have only one group who is trying to deal with redox state analysis, maybe not in a completely correct way, but still it's trying to do something. And we have another group who's trying to look at how pH can change something. So, but we do have a lot of other possibilities. We have salts, we have temperature, and none of this stuff is not taken into account. And I think, and you know, so it will be coming back to extremophiles. So we have some organisms which can survive in boiling water, so pH 2, with very high concentration of salt. Do you believe that organism which is sitting in this boiling water with pH 2 and very high salt condition, or pH 11 and very high salt condition, so you have exactly the same requirement for survival from viewpoint of amino acid sequencing protein? And the answer will be no. So, but we don't have any computational tool, so which would be able to look at this stuff. And counter argument will be we don't have too much data to train our computational tools. So, so to deal with all of this stuff. So it's kind of catch-22.
Yes. Thank you.
So, other questions?
Are there any other questions? The chaos theory you mentioned, you know, we had looked into it in the past with Orchide. It's a, you know, very well founded nonlinear dynamics theory. You know, when you say chaos theory, is it in that sense you are analyzing the motion of the protein structure, or is it more like you mean chaos? You know,
it's chaos. So, you know, entire biology is chaos. So we have structure which is originating from chaos. So
Yes, but
definitely,
I mean, when you say chaos in nonlinear dynamics, it has a specific connotation. You know, you mean
yeah, I know.
it's a
Yes.
dynamical system.
And I mean it. Yes. So
Yeah.
definitely. So it will be coming into complexity. Yes. So complex system is not a simple sum of components. It's more sophisticated. And proteins, many proteins will be not completely chaotic systems, so they will be edge of chaos. So which means also sitting somewhere in boundary. And actually it will define why they're so extremely sensitive to changes in environment. So we just put a paper which we are trying to publish, talking about intelligence in proteins. So proteins actually they do have some rudimentary, some primitive intelligence. So it's not in a way how you will use intelligence in human being. So, but it's definitely there. So they can memorize, so they can change environment, they can accommodate to environment. So they definitely do have features which are ascribed to behavior of semi-intelligent systems. And it will be coming back to complex system. It will be coming to everything else. Yeah.
Unless there are no other questions, Vladimir, it's been a pleasure to listen to you, and we hope, you know, to continue discussions later on in this regard. And thank you for
Sorry, I took more than half an hour. So, but you can
no,
cut whatever you want out of this.
That's
stream.
great.
that's okay. You know, it
Great.
was very interesting. You know, you know, you presented a lot of information, you know, in a relatively short period of time, and it was very beneficial for us. Thank you.
Thank you.
Thank you.
Yeah, thank you.
Thank you. So hope to see you next time. Have a nice day.
Absolutely.